talk is entitled Optimizing Spray Drying Strategies for Commercial Pharmaceutical Manufacturing. My name is Mira, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Catalent, who developed the content for this presentation. Catalent is a global leader in providing integrated services, superior drug delivery technologies, and manufacturing solutions to help life science innovators develop and launch successful pharmaceuticals, biologics, and consumer health products. Catalent has built an advanced range of technologies and capabilities and are dedicated to enabling a better and faster customer journey from product development to patient. With over 85 years of experience in the industry, Catalent supports our partners in introducing 200 new products and manufacturing over 70 billion doses each year. At Catalent, we share a common goal: to put patients first and help people around the world live better and healthier lives. Now, I'd like to introduce our speakers for today. Our first speaker is John Scrafford, has worked at the Catalent San Diego site for more than 6 years. He began his career at Catalent as a process engineer and now manages the engineering and process improvement groups. He has expertise in process development, troubleshooting, and scale-up of solid oral dosage forms, including blending, milling, wet or dry granulation, compression, coating, and spray drying. John holds a bachelor's degree in chemical engineering with a minor in business from Northeastern University. Ryan Minikis is currently principal at ArcLight Pharmaceuticals, LLC, an independent consulting firm focusing on drug product development. Ryan previously held the position of Senior Director, North America PDS for Thermo Fisher Scientific, formerly Patheon. Ryan also held positions at Aggear Pharmaceuticals and Bend Research. Ryan's formative work experience included assignments in analytical and formulation development, bioanalysis, and quality control. And now, without further ado, I'd like to hand the mic over to our first speaker. They may begin whenever they are ready.
Okay, good morning, everyone. My portion of today's webinar will be focused on spray-dried dispersion scale-up considerations. First, we'll briefly cover the DCS, amorphous dispersions, and go over a spray drying process diagram. Then we'll discuss the roadmap for early development work and scale up through phase 2 clinical manufacturing. Finally, we'll touch on the quality by design framework and how it relates to scaling up spray-dried dispersion programs. So no spray drying webinar would be complete without a reference to the Developability Classification System, or DCS. This is a tool for formulators to understand the biopharmaceutical challenges of their drug candidates and develop a formulation pathway to address them. As we all know, a greater and greater percentage of drugs coming from discovery have solubility issues, meaning that we're seeing lots of DCS2 molecules. In the case of DCS2A molecules, these solubility issues can be overcome by increasing their surface area through techniques like micronization. If the drug candidate falls into DCS2B, however, other methods are needed to improve bioavailability. One of these methods, and the focus of today's presentation, is the generation of an amorphous solid dispersion. The figures here represent a crystalline dispersion and an amorphous dispersion. The crystalline form of a drug is a stable, low-energy state, which may cause solubility challenges. An amorphous drug is in a higher energy state, which provides improved solubility and thus the bioavailability enhancement we need for those DCS2B compounds. An amorphous dispersion is characterized by random order of the amorphous API inside a polymer system. The polymer stabilizes the amorphous API by physically separating the molecules, and this allows for complete release of the drug without inducing recrystallization. Hot melt extrusion and spray drying are the two main methods of generating an amorphous dispersion, and today we'll focus on spray drying. This is a process flow diagram of a typical pilot scale spray dryer, which operates in a closed loop system. On the left side, we have our feed solution being pumped in and atomized through a nozzle into a fine mist. The hot process gas comes in through the top of the chamber, the red arrow, and vaporizes the solvent coming out of the nozzle, leaving behind dried SDD particles. It's important to note that though the process gas coming in is relatively hot and referred to as the inlet temperature, the vaporization requires a large amount of energy, meaning that the SDD particles are mostly experiencing a much lower temperature, normally referred to as the outlet temperature. From there, the gas stream and the SDD particles continue on to the cyclone, which works like a vacuum cleaner, where the particles are basically heavier than the air and spiral down the sides of the cyclone and into the collection vessel. From there, the process gas stream goes up through the middle of the cyclone and then through a bag or cartridge filter where fines can be collected. As long as your particles are big enough, you should have very little material making its way past the cyclone and into the secondary collection vessel. The process gas stream then goes through a condenser where the majority of the vaporized solvent is removed from the gas stream and collected. The now dry process gas is then recycled through a fan box and heat exchanger and sent into the chamber, which closes the loop. So now let's discuss what a typical early development roadmap might look like. For a new candidate drug compound, the first step is to characterize the API and determine the DCS classification. For the purposes of this presentation, we're going to assume a DCS-2B compound and that spray drying has been selected as the technology platform to improve bioavailability. The next step will be conducting solvent screening to find a solvent system which provides both solubility and stability. It's important to consider the solubility of the polymer as well, which often necessitates a binary or even tertiary mixture of solvents. Acetone is very common, as is a combination of DCM and methanol. You want to ensure that your drug is stable in the spray solution, and preferably for several days, and also that your solids content is not so low that the process is inefficient. You will also want to evaluate different polymers. Your choices may be limited or directed to certain polymers based on the properties of the API and the chosen solvent system. You can make small batches of SDD with the various polymers and then compare those for kinetic solubility, stability, and physical properties to help narrow down your choices. From there, you still may nominate 2 to 3 SDD formulations for further evaluation, making enough material to do further physical and chemical stability analysis, as well as in vitro and ultimately in vivo performance testing. The goal coming out of this phase of the development process would be a physically and chemically stable amorphous SDD, which shows improved bioavailability and performance. Most of this early formulation development work will be conducted on a lab-scale spray dryer. There are many options out there in the industry, with the most common being the Buki B290, which many of you are probably familiar with. Another great tool for these early development runs is the Procept 4 matrix, as it can run very small batches on the order of a couple grams and also has additional process controls such as drying gas flow rate set points and precise atomization controls. These first two options are fine for lab work, but with all of the glass pieces and the inability to run for long periods of time, they sort of fall short for larger batches. The GEA SD Micro is a good option for those larger development batches or even small GMP batches, as it is able to run continuously as opposed to the other lab units, which must be paused to collect product. It does this with a manual butterfly valve underneath the cyclone. When it comes time to actually scale up from a lab scale unit, you can do the mass and energy balance to determine your starting points. However, the particle characteristics are really the most important thing. In other words, you want to make sure that you are matching or even improving the product characteristics, not relying solely on the mathematical model. But as an example here, we are showing the case of a scale up from an SD micro to a mobile miner. And so here you're shooting to keep the particle size and residual solvent level the same, and the solids loading will stay constant because we've already locked that in during formulation development. From there, you'll maintain the same outlet temperature in order to keep the morphology the same, and then you can account for the higher drying gas and feed solution flow rates in order to determine your new inlet temperature set point. So just to reiterate that point, the biggest piece of the puzzle is determining the correct inlet temperature at your given spray rate, which will maintain the outlet at your desired temperature. Similarly, you can do the math on the atomization parameters to maintain the particle size. Note that the increase in atomization error is not linear, which may become problematic with increasing scale. The equipment most frequently used at pilot scale is the GEA Nyro Mobile Miner, or PSD-1, which are used at Catalent in San Diego and Nottingham. This spray dryer operates continuously in a closed-loop system. To do this, it has automated butterfly valves, which allow the collection of product while the machine continues to run. In terms of throughput, it operates in the range of about 6 to 11 kilos per hour of feed solution, depending on the solvent system that was selected during development. And I've done enough of these webinars to know that you're currently wondering what the minimum batch size is. And the answer, of course, is that it depends. It will depend on things like the solids content, the solution viscosity, the spray rate, and so on. But my rule of thumb is to spray for at least 30 minutes to allow the system to equilibrate. So if you assume 10% solids and 120 grams per minute as your feed rate, then you would need roughly 3.6 kilos of feed solution, which would generate 360 grams of SDD. You can then back calculate to determine the required amount of API based on the drug loading for your program. The scale-up example we just looked at assumes the use of a two-fluid nozzle, which are often used on lab-scale spray dryers. However, larger spray dryers generally use high-pressure nozzle systems, and these differ in a number of key areas. So two-fluid nozzle systems use a standard peristaltic pump for the feed solution and use nitrogen gas to break up and atomize the liquid stream. The ratio of atomization gas to liquid flow rate is really the key. So increasing the liquid flow rate creates larger droplets and thus larger particles, while increasing the gas flow rate breaks up the liquid stream further, creating smaller droplets and thus smaller particles. With high-pressure nozzle systems, there is no atomization gas. Instead, they use a high-pressure diaphragm pump to push the feed solution through a very small orifice, generally between 0.1 to 0.4 millimeters wide. And that is very small, but it's actually right here. And by putting several hundred psi of pressure behind this nozzle orifice, we're able to create a nice conical spray pattern that is very consistent. One key thing to note is that with higher feed flow rate, the pressure increases, which in turn decreases the particle size. And really, this is the opposite effect of increasing the feed solution rate on a two-fluid nozzle. So in short, you want to use the largest nozzle orifice you can at the lowest pressure, which still achieves proper atomization. And this should be the best balance of feed rate and resulting particle size. So in comparison, two-fluid nozzles do allow for more control over the absolute particle size because you have the extra lever of being able to adjust the atomization gas. But pressure nozzles have a tighter distribution and are really the primary nozzles used at PSD-1 scale and beyond because they do not require nitrogen gas, which, remember, scales exponentially with increasing feed rate. So in short, we generally move programs to a pressure nozzle at the first opportunity, since that is likely where the project will end up eventually. So even after the spray drying process is complete, the SDD will still have some amount of residual solvent, usually in the range of about 2 to 10%. And this must be driven off through secondary drying in order to meet regulatory guidelines. One thing to note is that the values shown here are the amount of residual solvent allowed in the final product. However, we will typically ensure that the SDD on its own is below these limits, and that diluting it during final dosage manufacturing is an additional safety factor. You'll also notice that the limits for some of these are quite low, and if you're unlucky, your drug may preferentially cling to some of these solvents. So you'll want to be sure to factor that in to your solvent selection during your early formulation development work. Ultimately, you'll need to complete drying studies to determine the length of drying required, but a realistic goal is often to achieve overnight drying, though that's not always possible. In these drying studies, you will pull samples for GC testing at various time points in order to build a drying curve, and that will help you to predict the total amount of required drying time. In terms of equipment options, at small scales, vacuum ovens with trays are typically used, and you can opt to use heat and/or nitrogen purge to aid in the drying process. As you increase in scale, a forced air convection oven may also be used, such as a Gruenberg. These ovens use a constant flow of heated air over the powder bed to help drive off the solvent as opposed to a vacuum. Another great option is a rotary vacuum mixer, such as those made by Ikato, which combine vacuum with kinetic mixing. It's basically a planetary mixer under vacuum. Rotary vacuum dryers are likely the best option for later stage and commercial programs due to increasing scale and the often shorter drying duration. So at this point, we've discussed many of the options and decisions you'll make as you progress through the development process. And now we'll talk about the framework that you can use to do this. So many people have heard the term quality by design, and on the surface, it sort of seems obvious. Of course, you're going to put quality into your product. So what is the FDA really asking you to do? And I think ultimately, they're asking you to use the following framework in your decision-making as you progress through the development process. One of the terms you'll hear is Quality Target Product Profile, or QTPP. And this is a prospective summary of the quality characteristics of a drug that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of the drug product. So really, I think of the QTPP as a summary document, or even really just a table, which will list out all of the characteristics that you require for a successful drug. It's important to note that you could have a QTPP for your final drug product and also a separate QTPP for your intermediate, in this case, your SDD. Another common term would be the critical quality attribute, or CQA. And this is a physical, chemical property or characteristic that could be within an appropriate limit, range, or distribution to ensure the desired product quality. So really, these are the individual attributes that will make up your QTPP. A critical process parameter, this is a key variable that are monitored to detect deviations in standardized production operations or changes in critical quality attributes. So really, these are just the variables and the parameters which will ensure that you meet your CQAs. So overall, quality by design is the application of this framework from the beginning of drug development, wherein decisions are made with these elements in mind. So let's go through some examples. There are many CQAs for a typical spray drying process, so we'll touch on just a few here. First, the physical state of the drug is critical, and obviously we are looking for an amorphous solid dispersion indicated by a single TG during DSC analysis and an amorphous halo shown via PXRD. So these might be specific line items on your QTPP. Other examples are the API to polymer ratio and the amount of residual solvents in the SDD. These can have an impact on the physical stability of your dispersion and thus are CQAs. Another example might be the particle size of the SDD, which may have an impact on the dissolution, performance, and downstream powder handling. Each of these CQAs will have CPPs associated with them, and in this last case, you'll be looking at things like the spray solution viscosity, the spray rate, and the atomization parameters to ensure that they are within an appropriate range to maintain your target particle size. There are many other CPPs for a typical spray drying process, but let's discuss particle drying as another example. The drying will control the morphology or shape of the particles, the density, and the residual solvent level of the SDD. The drying is a function of the inlet and outlet temperatures, as well as the residence time of the particles in the chamber. So generally for amorphous dispersions for solid dosage manufacturing, we're shooting for shriveled raisin morphology, shown here on the bottom left. And these are relatively dense, they have better flow properties, and are formed by drying at lower temperatures and thus more slowly. Spheres, shown in the middle, are generally bulky and difficult to work with during downstream processing, and are generally the result of drying at higher temperatures and thus more quickly. If you set your inlet temperature to the scorched earth setting and are drying at ludicrous speed, you may even burst those spheres open, which creates even bulkier material and more issues with downstream processing. And yes, those are the officially accepted industry terms, so make sure to jot those down. The general rule of thumb is that if you want the outlet temperature to be relatively close, and most likely below the boiling point of your primary solvent. Remember that the various solvents have not only different boiling points, but also different heats of vaporization. And these both play a big role in determining the optimal outlet temperature, which may be slightly different for each program and each solvent system. There are, of course, many other attributes and parameters for a given STD program. However, not all of them will be considered a CQA or a CPP. It will depend on the program, but some of them may only be related to processability or efficiency. For example, the flowability and compressibility of the STD, which are mainly controlled by the drying temperatures, may not have a performance or quality impact on the STD itself, but it could be very important for the ease of downstream processing in the form of uniformity or compression issues. Other examples might be the solids loading of your feed solution or the drying gas flow rate. It's possible that these factors on their own may not have an effect on the performance of the product. However, they can make a huge difference in efficiency when you get to larger scale. Now, ideally, you have been using this QBD mindset from the beginning, and you already have an optimized product. As you get closer to later phase 2, phase 3, and commercial manufacturing, however, it is always pertinent to reconsider some of your previous decisions. The first is relatively obvious, but what equipment have you been making your clinical batches on, and how far will you need to scale up? Remember that going from a PSD1 to a PSD2 is almost a 4x increase in throughput. So depending on your clinical study design and dose strength, this next step may be a large increase in batch size with longer run times, or it may be that going to a PSD2 will actually result in lower yields if your batch sizes remain relatively small. You'll also want to consider whether your early phase process had any quirks which may be difficult to replicate at larger scale, such as complicated solution preparation or filtration. If so, consider simplifying or better yet, eliminating these steps before you do go to the tech transfer batches at larger scales. So at this point in your clinical program, you also may have additional information about your projected dose strength. And if it is higher than originally expected, this would be the time to consider increasing the drug loading in the SDI in order to achieve higher dose strengths. This higher drug loading would provide a more efficient process and smaller dosage unit size, but also could have negative consequences on performance or stability that will need to be checked. If you didn't do it earlier in the development process, evaluating alternative polymers and solvents may provide a path to improving performance and stability. And again, now would be the time to look at these before you scale up. Similarly, any improvement you can make in the solids loading of your feed solution or in the process parameters will pay major dividends in the form of a higher throughput and a more efficient manufacturing process. But once again, this of course needs to be checked and balanced with ensuring that there's no impact to particle morphology or downstream performance. The roadmap for any given program will differ. However, these are some of the key considerations to make before moving into tech transfer activities at larger scales. So here are a few key takeaways from the first portion of this talk. First, spray drying is a great option for bioavailability enhancement of DCS-2B drugs. In early development, you'll need to lock in your solvents and polymer and ensure that you are making a physically and chemically stable STD with improved bioavailability and performance. As you look to optimize your process, you'll want to make equipment selections, such as the atomization and secondary drying methods, determine your optimal drying kinetics and particle morphology, and also consider how to improve the efficiency of your manufacturing process, such as by increasing the solids loading. Ideally, you've used a QBD mindset from the beginning, and minimal optimization work is needed before scale up to late stage equipment, which will ultimately save time, money, and often most importantly, precious API. Now I'm going to hand this over to Ryan Minikis, who will discuss late stage program planning in greater detail. Thank you.
All right, thank you very much, John. Great primer on spray-dried dispersion technology, its assessment and its application in clinical development. As John said, I'm going to focus build on some of the concepts that John spoke about and focus on late-stage program planning for spray-dried dispersion product development programs. So starting with the concept of the quality target product profile and its role in late-stage prescription product development. So what I would propose is that the product development goal through all phases is ultimately to deliver a commercially viable prescription drug product. How we define commercially viable is probably beyond the scope of this talk, but there's well-defined or well-accepted attributes in the categories of safety, efficacy, quality, and of course, commercially relevant attributes like cost, shelf life, storage, and distribution. As John told us, the quality target product profile is a summary of the product's characteristics as they apply to these above categories. It's a guiding document for late-stage product design and development, and very importantly, it should have broad stakeholders in its development and specification. These will include the clinical and non-clinical groups, as well as supply chain, quality, regulatory, and as you approach commercial, marketing as well. We should consider ranges and contingency values when we build our QTPP during development. So for example, what if the unit dose needs to double in a pivotal trial? I think having a contingency for that is a prudent example of a range or contingency. Also, I want to build on what John mentioned is the spray-dried dispersion as a drug product intermediate. What that means is that it's a functional component of your final product, most likely a tablet or capsule, but could be other dose images as well. And what I'd like to propose here is that we start with building the finished product quality target product profile, and from that derive the SDD QTPP. So to hit very high level on what a final product quality target product profile might look like, we have a table here, and this again is for a typical prescription finished product. I've proposed that in your table you'll have dosing route, so oral, nasal, lung, and so on. Most applications of spray-dried dispersions are going to be oral. You're going to specify the unit dose format, so for example, tablet, capsule, sachet, orally disintegrating, or so on. Unit dose image, this is color, shape, size, embossing, and other elements of the appearance of your product. Unit dose strength, oftentimes we deal with multiple strengths. There's the need for placebos during controlled clinical trials. Sometimes we have fixed dose combinations, and of course dose uniformity as well. There's also dosing frequency and pill burden. As we get into these topics, you'll come into topics like FDA inactive ingredient or GRAS limits on certain components of your dosage form, excipients. And are you intending to treat special, pediatric, or other compromised patient populations? There's also performance of the drug product, which the whole application of the spray-dried dispersion technology is typically towards solubilization, but could also include things like modified and controlled release, specific tissue targeting, or specific pharmacokinetic performance parameters. There's always the topic of shelf life and long-term stability and its packaging, and then finally distribution. So for example, secondary packaging, kit format, mechanical robustness, and ultimately global markets. So once we've written down and have determined our finished product characteristics, as shown on the previous slide, I believe we can derive what are appropriate SDD attributes. So for dosing route, things like excipient choice and levels, also microbiological quality. For format, we'll have attributes like flow, excipient compatibility, compressibility, and wettability, all that impact downstream processes like tableting. Unit dose image, drug loading, color compatibility, compressibility, sticking. Unit dose strength, again, drug loading, the potential for multiple drug product intermediates, common granulation approaches, dosing frequency, again, drug loading and release rates, pill burden is, again, drug loading, and then also percent STD in your final formula, whether it be a tablet or capsule. We have performance attributes, which, again, can implicate drug loading, but also polymer choice, the extent of solubilization, its physical form and stability, its particle size, and specific surface area. Shelf life has long-term chemical and physical stability requirements. We also have to consider the bulk intermediates, shelf life, and functional packaging components. And then finally, for distribution, typically we'll demonstrate temperature cycling appropriate for the distribution routes. So here, I think one thing that becomes obvious is that STD drug loading is the single most limiting or alternatively enabling factor in spray-dried dispersion development. This determines things like what unit dose can be achieved. And so therefore, it's very important to determine early if your early phase product is going to need reformulation. In practical terms, this is going to be particularly relevant if you need a unit dose greater in the range or greater than 100 to 200 milligrams. So shifting away from building your quality target product profile for a spray-dried dispersion and also your finished product, there are scale-up, specific process scale-up considerations. John covered these as well, and I won't go into the specifics other than to reiterate that you can build thermodynamic models for the spray drying process. Here we have one that's showing scale up from a PSD1 to a PSD2, where we have maintained that outlet temperature, as John described to us, by varying other scale parameters on the equipment. On the left, what we have is that outlet temperature graphed as a function of specific drying ratio, which is the mass feed rate of the solvent over the mass feed rate of the drying gas, with contours showing our outlet temperatures and relative saturation of the solvent. And the box really represents what we believe is an operable space, and the dot in the middle actually represents our target condition. The other part of the process that needs to typically be scaled up, in addition to thermodynamic, is the atomization process. So again, John's talked quite a bit about the different atomization options, twin fluid and pressure nozzle, for example. In this case, I'm just showing a tool. In this case, a Malvern Spray Tech that's analyzing atomization plumes from various nozzles. And what we have on the left here is a contour plot showing us multiples of PSD1 flow rate on the x-axis and atomization pressure on the y-axis, with contours showing us where the mean droplet size distribution will lie for different nozzle orifices. Again, the whole idea here is essentially to create a lookup table, or curve in this case, that will allow us to pick the correct process parameters for initial applications at scale. So once we have these modeling done, we can then go ahead and define our process design space. This work's primarily done in silico. At this point, we can create a risk assessment. We can identify our most likely significant factors, otherwise known as critical process parameters, and our significant responses, otherwise known as our critical quality attributes, perform a design of experiments, and then ultimately analyze, statistically analyze via ANOVA, and interpret that DOE. The best interpretation of a DOE would result in things like normal operating ranges and proven operating ranges for our product. In this, I think important considerations are whether or not the sponsor or the vendor is driving this modeling exercise and the subsequent experimental design, analysis, and interpretation. What the scale of the equipment is that will be used for the experimentation and the ultimate application, the material requirements for performing the experiments. In some cases, we'll use surrogate where API is limited. And then finally, are there any required applications of the material that will come out of these DOEs? Moving on from process scale-up, we have the topic of vendor selection. So in the current market supply situations, most sponsors, whether big pharma or biotech, will be evaluating external CDMO suppliers for late phase and commercial spray drying. Many of these late phase and commercial spray drying manufacturing will require a site tech transfer to a larger equipment platform. So in other words, oftentimes scale up also comes with site transfer. In this project scenario, we have the sponsor or the innovator company, and we will have a sending unit and a receiving unit. And the diagrams on the lower left represent two different options for how to approach this problem. On the left, we have the sponsor interacting with both the sending and receiving units, and an open line of communication between the sending unit and the receiving unit, versus the option on the right, where the sponsor is the intermediary between the sending unit and the receiving unit. This will have implications on things like service contracts, confidentiality and IP, quality management system requirements and compatibility, import and export and trade compliance for materials distribution, analytical method transfer and validation, and perhaps most importantly from the sponsor's perspective, the project management and technical burden that will lie upon that organization. For vendor selection, we have, we can look at site TT project scenario 1. I think we probably could all agree, barring business complications, that having a direct line of communication between the sending unit and the receiving unit is our best chance for success. Within that scenario, we can have two different situations. In the first scenario, or in the first situation on the left, we see that the sending unit is CDMO company number 1, sending the product to CDMO company number 2. And on the right, we see that the sending unit and the receiving unit are part of the same CDMO company, but different sites. This introduces another set of considerations for the sponsor organization, including contracting and subcontracting complexities, the technical capabilities and talent and experience, sometimes product-specific experience of the organizations involved. Again, materials, logistics, and trade compliance, local EHS requirements and alignment between the multiple organizations involved. So this can get to topics like requirements for containment and different occupational health implications, quality system requirements and alignment between the organizations. Again, material qualification requirements, so materials can flow between the different sites. Again, global program management versus site project management. And again, reemphasizing that the choices that you make in this scenario, or between these two scenarios, could have a significant implication in terms of project management and technical burden that's placed upon the sponsor organization. So again, once we select a scenario and we select our vendors, again, going with scenario 1, where we have the sending unit interacting directly with the receiving unit. Now we actually have to come up with an appropriate program management and governance approach. On the lower left-hand side, I'm demonstrating a swim lane diagram that pictorially represents both, several important aspects of program management. First of all, this is the guiding document for global program governance. Second of all, it should allow for clear points of communication between the organizations and clear responsibilities for each organization. And finally, clear timelines and deliverables. Of course, other tools like Gantt charts will be important to capture the level of detail required. I think it's especially instructive to start with something like this swim lane diagram to make sure all the organizations understand their role and their responsibilities in the tech transfer process. Another topic in terms of scale up and tech transfer is material requirements and planning. So the first point I'd like to emphasize to the audience is that in the case of spray-dried dispersions, you are developing an entirely separate drug product intermediate manufacturing process. Because of this, you're introducing additional unit operations to the manufacturing process, and the expectation should be that more API will be required versus a conventional dosage form. In my experience, API availability is almost always a limiting factor for scale-up and tech transfer activities. It will determine what experiments can be done, where surrogates will need to be used, and where interpolation and extrapolation need to be used in lieu of more experimentation. When you're planning your material, use rational, product, and process-specific material planning approaches. So I'd encourage you to avoid phase-based rules of thumb. So for example, I need 5 kilos for phase 1 and 20 kilos for phase 2. I see that as a way that customers often get themselves into a pickle with respect to material availability. So I encourage you to work early and often with your clinical teams as well as your vendors to understand what these requirements are going to be and plan well as far in advance as possible. We have API and drug product CMC teams collaborating again early and often. And one of the biggest topics that I think that you should consider when you're looking at scale up as it relates to material use is representative run times for your scale up and DOE work. So for example, 10 minutes may be adequate to obtain representative product observations, things like particle size, morphology, and residual solvent. But you need at least an hour runtime to observe get a representative observation of the process. So things like yield, chamber buildup, nozzle bearding and clogging, and cyclone bridging. So make sure that you understand what your objectives are for the specific experiments and that you have adequate material to demonstrate those objectives. Finally, consider the materials use resulting from the scale-up and TT activity. So oftentimes a tech transfer batch may also be a clinical batch, in which case you would need GMP controls for that development run. Also product testing requirements, downstream process development needs. So for example, development of the tablet or capsule filling process, and then also bioequivalence and PK testing requirements. So for example, if you change a formulation during clinical development, you may need to run a relative bioavailability study to support that change. Key takeaways. So again, remember the product development goal is a commercially viable prescription drug product. Start with the finished product quality target product profile and derive the spray-dried dispersion QTPP. Consider scale-up and TT program technical approach and requirements. This includes in silico modeling, process design space, and scale-up DOE work, and also talent and experience. During vendor selection, consider the business complexities and the project management and technical burden that the sponsor organization may be able to or willing to take on in that process. In terms of tech transfer program management, create clear program governance with clear organizational roles and responsibilities, timelines, and deliverables. And in terms of API material requirements, develop a rational and product-specific supply plan considering SDI as a separate manufacturing process. Thank you very much. I'll turn it back to for Q&A.
Thank you very much for that insightful presentation. Now I would like to invite our audience members to continue sending their questions or comments right now using the questions window for the Q&A portion of this webinar. I've already received some of those questions, so I'll begin with those. Our first question here today is, for nasal spray use, small size droplets with high pressure have better bioavailability. What is the utility of using larger size droplets with low nozzle pressure?
Yep, I can take this one. So the recommendations earlier were made regarding using a larger nozzle orifice with low pressure were really intended for amorphous solid dispersion programs going into a solid dosage form, like a tablet or a capsule. where in most cases, larger particles provide improved performance and ease of downstream handling. If your program is targeted at nasal inhalation, then certainly generally smaller particles provide improved performance. And in that case, you would adjust the atomization parameters to make smaller droplets. So as the question stated, that's when you would be looking at using higher pressure in a pressure nozzle to again, generate those smaller droplets and thus smaller particles.
Thank you very much. Our next question here is, based on your experience, are there process parameters that would allow avoiding a secondary drying, i.e. achieve drying task low residual solvent only with spray dryer?
I'm happy to take that, John. Based on my experience, it's unlikely that you are going to run a spray drying process that is going to achieve the ICH limits of solvents, as John showed in one of his slides, straight out of the spray dryer. It's more likely that you're going to require secondary drying. Much more likely that you're going to require secondary drying. If you ran the process as hot and dry as may be required to yield product that doesn't require secondary drying, I think you'd most likely have deleterious effects on the product properties. So you would be drying at ludicrous speed, essentially, as John referred to it, and you'd probably end up with things like shattered particles, very fluffy powder, static problems, and whatnot. It may be possible, but again, it's unlikely. And that's from organic solvents. With water, the situation is different, and spray drying from water is relevant for some products. In that case, I think it's going to be very product specific, and you may be able to achieve products without a secondary drying process being needed.
Thank you very much for that answer. Our next question is, solutions with polymers are often viscous and may clog the nozzle. How do you handle that in high pressure nozzles where the nozzle orifice is substantially smaller, thus a higher chance of nozzle clogging?
I can take this one. So it's a great question, and it's really a very common issue that needs to be dealt with. So I guess the first recommendation would be to ensure that you have a robust solution preparation process where the polymer and the API are really completely fully dissolved. And so the biggest risk is having any undissolved material in the feed solution, whether that's polymer or even some extraneous matter, which can really easily clog that small nozzle orifice. So a common method of addressing this, certainly with robust mixing and having the appropriate mixing time, but additionally you can utilize a stainless steel mesh filter in the feed solution line, and that's usually sitting right before the pump. And so these filters range in size, but generally between 100 to 200 microns, and they should prevent most of that material from getting to the nozzle. Even still, nozzle clogs will happen occasionally, and so it's really important to really keep a pair of eyes on the equipment to ensure that the spray pattern is appropriate and things like the atomization parameters and the feed rate stay consistent. So that really as soon as a nozzle clog is observed, then you can take action. And really it's kind of one of two ways you may do that. You may switch to a pure solvent feed in the nozzle, or you may pause the spray to remove the nozzle for cleaning.
Thank you. Our next question is, apart from SDD, have you ever done microencapsulation using spray drying?
I'm happy to take that one, John. I've used that application a few times and well aware of plenty of precedent in the literature for microencapsulation. It's typically a different type of application than spray drying amorphous dispersion, as the asker implies there. In this case, you're looking at encapsulating some material, usually an API, inside of another material, usually a polymer, that may have different sorts of properties for either wetting or release rate. So PLGA is an example. Again, I'm aware of those, typically not for solubilization, and more typically for niche applications like, so for example, for a depot injection, for example, in an aseptic spray drying application is an example where I've used that technology.
Thank you. Our next question here is, as drying carried out at higher temperatures, how are safety aspects considered?
I can take that one. So really, when using organic solvents, the required temperatures are a bit lower, with the inlet temperature generally ranging about 90 to 130 degrees C. When spraying with aqueous feed solutions, the inlet temperature may be all the way up to 200 C. So this certainly does present some safety challenges. However, these usually can be mitigated with insulation on any of the exposed piping in the equipment. You also have to remember that there's a large amount of heat that's dissipated in vaporizing the feed solution. So the bulk of the equipment is at a much lower temperature. So that's that outlet temperature. It's usually in the range of maybe 35 to 60 C for organic solvents and 90 to 100 C for aqueous sprays. So there's sort of generally a limited amount of the equipment that needs that insulation. But really, I think the biggest risk is around, if an operator was to touch one of those areas of piping that is exposed. So usually with insulation, we can mitigate that.
Thank you.
I'll add to that, John, if you don't mind, is that, I just want to reemphasize to everybody that's listening that these are well-established technologies, and there's several vendors that make spray dryers that are safe and rated for solvent use. It gets into some fairly specific detail, like auto ignition temperatures of the solvents that are being used and sheath temperatures of heating elements and whatnot. All I can say is and want to emphasize to the listenership is that engage a good engineering firm and a reputable supplier, and they should be able to help you with these. Don't necessarily try to go it alone in terms of equipment design for that safety reason.
Thank you. Our next question here is, so many developmental compounds these days are using SDD-based formulation. This makes it challenging to conduct human mass balance study with radio-labeled compound. This is particularly challenging with low solubility compounds since alternative formulation is not feasible. Do you have any experience or suggestions in this scenario?
I'm happy to take that, John. I'm familiar with the challenge that the listener is alluding to. Basically, it's difficult to spray radioactively labeled compounds via this process in a way that's safe to the operator and can be cleaned up and contained afterwards. In cases where we've needed to do radio-labeled studies with amorphous dispersions, I've used different process technologies to get to effectively functionally similar materials, and luckily typically you only need gram scales. So examples include rotary evaporation with subsequent milling steps, or even extrusion as a potential alternative to spray drying radio-labeled substances. And so again, I think the way that I've solved that problem in the past is to look at small scale, lab scale process alternatives to spray drying to yield similar dispersions of drug and polymer.
Thank you very much for that. Our next question is, what are the SDD specific considerations when making decision on the final dosage form, capsules versus tablets?
Yeah, I can start there. So, first of all, with SCD programs, you can certainly go into both capsules and tablets. It's certainly not, directed at a single dosage form. I think the main considerations are going to be around the compressibility of the material and the tendency to gel. So depending on the polymer that's used, as well as the amount, the loading of the polymer, gelling issues can often be seen during dissolution, affecting performance. So, when you're using in a capsule formulation, you're not compacting the material as much, and that could have some effect. versus a tablet, depending on the level of compression, that could actually hinder, or in some cases, in many cases actually, can improve the dissolution of the STD as well. So I think it's really mainly around the compressibility of the material coming out of the spray dryer. That's going to be dictated by things like the particle morphology and the density, as well as the key consideration being the polymer loading and how that's going to affect any sort of gelling issues. Anything else to add, Ron?
Yeah, I think all I would add to that is the compression itself. When you use a tablet, obviously allows for a little bit more material in a unit dose. So what I mean by that is in a size OT or double OT capsule body, you can typically fill maybe 500, 600 for denser granulation, 600 milligrams of material. Whereas in a tablet of a similar, I guess, aspect ratio and size to that size OT or double OT capsule, you can probably get closer to a gram of material. So for very large, higher dose products, a tablet may be more practical if you're looking at, pill burden issues like number of pills per day for the patient.
Thank you for that. Our next question here is, how much maximum percentage solid load can be considered?
I can talk to that, John, if you like. Typically, solids loading, I like to think that a commercializable process, reasonably efficient commercial process, will be at least 10 weight %. That may not always be the case, and you may need to make concessions there based on solubility. I've not seen any cases for STDs that go above 20%. And that usually is limited by the viscosity of the polymer. So key in that is going to be some viscosity experiments. And again, those experiments on atomization pressure or orifice size to determine what your limit of atomization is. But in that range, I think 10 to 15% is considered a good commercializable process. Below that, you're making some efficiency and probably cost concessions. Above that, you're going to have atomization challenges due to the viscosity of the polymer.
Thank you very much. Our next question here is, do you have any specific recommendations for spray drying a product for sachet dosing?
Again, I'm happy to take that, John, if you like. Again, depends on the application of the sachet dosing. Is this something that you're wanting to formulate in a way that's, extended release, or perhaps you want to access different types of patient populations with swallowing issues? For example, pediatric populations, or even people with they're likely to have disabilities and swallowing issues, where a sachet and a drink type presentation is better than a tablet or capsule. In those cases, I think John alluded to gelling. That's definitely one tendency of the polymer that you're going to want to take into consideration, and its ability to essentially its dispersibility in the aqueous matrix. You're also, I think, and oftentimes this is added to the reconstitution medium and not the formulation itself, but some sort of viscosity modifier like agar gum or xanthan gum or cellulosic that is going to modify the viscosity and help the suspendability and recovery of that material from the sachet drink, essentially. So things that are going to keep those particles in suspension for timescales that are relevant to dosing. Again, gets into specifics, but it starts with really considering what the attributes are that you're looking at in terms of the dosage profile and the patients that you want to dose with it.
Well, thank you very much for your answers. We've now reached the end of the Q&A portion of this webinar. If we couldn't attend to your questions, the team at Catalent may follow up with you, or if you have any further questions, please direct them to the contact information that is now showing on your screens. Thank you for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Now, please join us in thanking our speakers, John and Ryan, and feel free to share the recorded version of this webinar using the link in your chat box when it becomes available to you. We hope you found this conference informative, and have a great day, everyone.